All right, good afternoon, everybody. We'll get started soon. Welcome to this week's virtual collection tour. This week we'll have Patrick walk us through what in the world is herbaria and why do we have them. So think about the word herbaria for real quick and try to predict what it is and how do you think scientists like the one at the Peabody study it. Just as a reminder, feel free to use the chat to make any comments that you have, but also don't forget that there is a Q&A section for you all to drop any questions that you have that you might want Patrick to answer. So again, use the chat to make any comments that you might have, but also don't drop your questions in there. Drop it in the Q&A section. So I hope you guys are all excited for today's tour. And now we have Patrick.
Hi, so welcome to the Yale University Herbarium. So my name is Patrick Sweeney, and I'm the collections manager of Yale's Herbarium. And today I'd like to give you sort of a brief introduction to Herbaria, so that's plural for herbarium. And I'll use Yale's Herbarium, singular, as sort of an example of a herbarium. So what are herbaria? So really herbaria are it is the botanical collection for the Peabody. So it's a botanical natural history collection. And the Peabody's herbarium, we refer to it as the Yale University Herbarium, has about 350,000 items from all over the world, dating back to the 18th century. But more generally, there are thousands of herbaria out there in the world. There are over 3,500 actually. And collectively, we have about 400 million specimens, 400 million items from all over the planet. And what I'll do now is sort of show you examples of what we have in herbaria and talk a little bit about plant diversity. And then I'll show you specific interesting things in our herbarium here at Yale that I think you'll like to see. So we'll start out with the spread I have on the table here. So I pulled out a few examples of specimens that represent what we have in terms of groups that we have in the herbarium and the way they're prepared and eventually stored. I'll talk about that later. So what is a herbarium specimen? Sometimes if you look it up, like a definition in like the Oxford Dictionary, it will be described as a press-dried plant specimen. And this is a press-dried plant specimen. So this is a herbarium specimen. So this is a fern. And you might notice the plant, it's more than just the plant. The plant's mounted to a sheet. And actually, all herbaria in the world use a standard size sheet. It's roughly 12 by 18. And on this sheet, we have the plant. And in addition, we have a label that tells us about the specimen itself. So this label will mention things such as what the plant is. It will show its Latin name, its scientific name. It will show where it was collected, when it was collected, who collected it, and it might also mention information about the habitat the plant was growing in, and it might mention things about the plant that you can't really see in the dried specimen, like if it was a tree, how tall it was, if it has flowers, what color the flowers are, because when the flowers get dried, the color can change, it often does. So that's sort of your typical herbarium specimen. And so these specimens represent many different groups on the tree of life, if you want to think about it that way. And we have plants, that's true, and that's mostly what we have. We have different groups. We have algae, so we have algae represented. We have things like mosses, and the mosses are prepared and sort of housed a bit differently than the fern you just saw. The mosses they're flattened, but they're sort of kept loose in a packet like so. So mosses, we have ferns and their allies. We have things like gymnosperms, so conifers. Here's a pine example here, and we do have the cones as well that go along with the flattened specimen on the sheet. And then we have flowering plants like the ones you see here. And so because this is a natural history collection, it's invaluable for research. And what I like to tell people is that for most species on the planet, most plant species, what we know about them is represented in the herbarium. Herbaria are kind of like databases, if you will. They have information about what the actual plant looks like. We have information about where it occurs, when it occurred there, information about habitat. The specimens themselves are information about what the plants look like. There might even be evidence of herbivory on the plants, things like that. And even the chemicals in the plants can be useful. For example, we can extract DNA from these specimens and build evolutionary trees for that kind of research. And this grass here, this specimen, which was collected in the late 1800s, you can actually see there are little chunks taken out of the leaves here and here. And those were used for DNA analysis. That was used in an evolutionary study that was published. And so all kinds of use. In addition to evolutionary studies, ecological studies, conservation, and education, much like the things we're doing here today with this tour. So lots of great uses. And in addition to plants, we have things that aren't plants. So historically, herbaria, before we had sort of the tree of life all figured out and knew what was what and how things were related, people were putting things in herbaria that we know aren't plants today. So for example, we have fungi, not many, but we do have fungi. And your typical herbarium usually has fungi. That's where they're housed. And we have things like lichens. And here's a lichen that was collected in Scandinavia in the 1970s. And it's actually still attached to the rock it was that it was occurring on. And so we have things like lichens, which you might know are actually two organisms. There's a fungus involved, and then there's a photosynthesizing organism involved, an algae or maybe a cyanobacterium. So more than just plants, but mostly plants. And so I hope you're getting a feeling for kind of what we have and how it can be important. And I like to put in a plug for plants. Here at the Peabody, the vertebrates, particularly the fossils, often steal the show. But I like to tell people that plants they are the backbone for life on planet Earth, right? If you think about it, from a human perspective, they provide our food, they serve various ecosystems or fulfill various ecosystem functions. They contribute to quality of life. Even the air we breathe is coming from plants. And so, understanding plant diversity, understanding things about individual species, such as where they occur, when they occurred there, what they look like, having DNA for these specimens for various things, other kinds of chemicals, all of that is eminently useful. And as I said earlier, for most plant species, that kind of information really is only present in these botanical collections in these herbaria. Maybe I'll pause for a second and ask if there are any questions.
Yeah, there's a couple questions.
Great.
Sarah asks, is the paper on the specimen, is it mounted on a special type of paper so it doesn't damage the specimen? And
Yes.
Does anything else go on top of it to further protect it?
Right, that's a very good question. So the paper currently is a special paper. It's a paper that we call archival quality paper. So this is a paper, it's usually a cotton rag paper, a very high percentage, usually 100% cotton rag, and it's a very thick paper. And that kind of paper over time won't become brittle. So if you think about old newspapers, maybe in an attic or old paperback books that you might have lying around, you might notice that paper deteriorates over time. It becomes very brittle and falls apart. These papers we use won't do that. So, and for that reason, we call them archival quality. And even the paper that the labels are printed on, the labels are the pieces of paper with the information. Even those are printed on special paper. We even consider the inks. And the glues we use as well are meant to last a long time and not interfere negatively with the plants. Likewise with the papers, there shouldn't be interactions between, say, the plant and the paper that are negative. In terms of putting things over them, we do put them in folders, in paper folders. You'll see that in a moment when we actually head back into the collection area. And they're put in cabinets in little cubbies, and these cabinets have seals, and the room itself is sort of a seal, if you want to think about it that way. But like plastic and things like that over the individual specimens, we don't actually do that.
So yeah, Diane asks, you don't need any gloves or anything when handling the sheets or collections?
No gloves. They're not handled so often that that's a concern. And we've talked with conservatives here at the Peabody, people who are really, who spend their lives dealing with conservation of natural history specimens, and gloves aren't recommended for the kind of use that these plants are put through, the kinds of uses.
Yeah, okay, thank you. Yeah, that's pretty much it.
And so, as I mentioned just a second ago, I mentioned going back into the collection room. This room we've been in is our work room. And so this is a room where we might actually study plants, or a researcher might visit and study plants. It's a place where we might database our specimens. So one big thing that we're doing to make our specimens more useful is we're imaging them. And we're taking information that's on that label I mentioned earlier and putting it into a database. And all that's going online to make these things useful for research and for education, so on and so forth. You might notice in this room that you'll see in just a second, we have imaging taking place right now. So we have a project focusing on imaging our some of our moss specimens. So we have a lot of things like this going on in this room. We have a library. The actually new specimens that are collected that come in are actually processed here and prepared and put on these sheets, as I've shown you. And so that's what happens in this room. It's our work room. And so what we're going to do now is we're going to head through this door. You'll head through the door following me into the room where we actually have our specimens. So this is the collection room. So as I mentioned earlier, we have about 350,000 specimens in this room. Most of them are flowering plant specimens. We do have a considerable number of mosses and their relatives. And we have a fair number of ferns and fern allies and gymnosperms, conifers. Not so many fungi, and just a few lichens. And most of these cabinets actually contain flowering plants. And so what I'll do now is I'll open some cabinets up and start looking at things and talking about them, particular items that'll give you more of a flavor for what we have. And then I've got some special things that I've sort of pulled out that I think you might be interested in seeing. So what we'll do first is we'll go to this cabinet. You get a feeling for how these plants are stored. So somebody earlier asked, are they covered? And I mentioned they were put in folders and put in cubbies and sealed away in the cabinets. This is how they're stored. So they're sort of stacked up in these folders, and they're arranged based on how we think they're related to each other at a high level. And then as we drill down, they're sort of arranged alphabetically by their scientific names and also by geography. So you might notice these colors in this cabinet, they represent different geographic regions. And so what I'd like to do now is just pull a folder out and show you some specimens sort of as they are in the herbarium. So here's a folder, and we have the family that the plant belongs in on the folder, that name, and then we have the genus of the plant. And if we open this folder up inside, we have a bunch of specimens that all belong to the same species. And this is a kind of primrose that occurs in the Western United States. And so I'm just pulling out the random specimen that's on top. You see, it looks like the specimens we saw out in the other room. It's mounted on a sheet. There's a label that mentions what it is, where it was collected. This was collected in Pueblo, Colorado in 1883 by R.D. Woodward. Not much more information here, but still very useful. We have the plant itself, which actually has information. I like to think about plants as being information. We have a flower. So we know this plant was flowering at this date. We have the leaves. There could be herbivory. There could be insect damage that has happened in the wild, and you could actually examine that on this plant. You can see actually here there might be some damage that was maybe caused by an insect or something. So, if you really study this carefully, you might be able to figure out maybe even what that insect was. And we have multiple specimens of the species in the folder. So here's another one collected by a different person in 1887. This one was collected in the San Juan Mountains in Colorado at 9,000 feet, so a bit more information. There's actually two specimens on this sheet. You might notice these have barcodes, and those barcodes were part of the digitization process. And so these specimens have been imaged and they've been put online. They've been databased too. Another specimen here. This is a bit later, 1935. So, I'm not going to go through the whole folder, but this gives you an idea. We have a whole stack of specimens here from this one species that occurs in the Western United States. The specimens date back to the 1800s, coming into the 20th century. And so we have information for this species in this herbarium about the distribution of this plant. We know when it occurred in different places. And we actually have the specimens themselves, which have all kinds of information associated with them. And so there's real power in having many, many specimens of the same species. And as I mentioned earlier, we're just one herbarium. We just have 350,000 specimens here out of this 400 million that are spread around the planet. So you can imagine collectively how these collections really are sort of encyclopedias of information about plants. And for many, many species, the information we have about them, it's in these collections and that's it. You're not going to open up, the encyclopedia and get all this information about their distribution, their morphology, what they look like. You're certainly not going to get DNA, things like that. So I sort of hope I'm giving you a feeling for why these are important and what we use them for. Any questions so far?
Yeah, there's a couple of so Helen asks for the lichen. How is it preserved? Is it virus killed, special process?
No, right. For the lichens, there is really no special process. They're often collected if they're not attached to a rock, and they can be detached from the rock or whatever it is they're growing on, maybe the ground or a tree or something. They can be they're not pressed flat. They're just dry. So they might be put in a low heat drying oven, like 95 degrees, very low humidity, and dried out over a few days. And then they might be frozen. And the freezing, we freeze everything that comes in to a quite cold temperature for a few days before it comes into this room or even the other room. Because one major thing we're concerned about in curating our collection are insects. Insects can come in, as you know, out in the wild. If you have a garden or something, insects love to eat your plants. Well, they love to eat dead, dried plants too. Different ones than you have in your garden. We don't have Japanese beetles here, for example. We have other kinds of bugs that eat our plants. There's actually a beetle that people have nicknamed the herbarium beetle, which is like, the arch nemesis of the herbarium collections manager. And so, if those get in your collection, they'll eat your dried plants, and it's awful, as you can imagine. And so freezing, very cold for a few days, it's very effective at killing eggs, larvae, and the adults. And so everything gets frozen. And so we freeze, the lichens would be dried, getting back to your question, at a low temperature, frozen, and then brought into the collection. And that's all the processing that would happen. There are no chemicals that are applied, nothing like that. So it's conceivable that some of these lichens could still be viable, potentially, I suppose, at least the ones that were collected more recently. And that's not a goal of ours. Everything in here is, we're not striving to keep things alive, but, it could happen. Was there another question?
Yeah. Anne says, I've seen a number of herbaria from the medieval Renaissance era in repositories overseas. And she was just wondering when and who set the standardized size and characteristics that Peabody has today.
Yeah, for the herbarium sheet size, that's a good question. And I think people have written a little bit about this, and I'm a little bit fuzzy on the history, but there were some very early botanists that were very important in the history of taxonomy, and sort of their practices became the standard. And that's sort of my understanding of it, if I understand your question correctly.
Okay.
That's sort of a brief, vague answer, but that's what I've got.
Yeah.
Okay.
Oh, do you want to take another couple questions or?
Sure, like maybe one more.
Okay. Sarah asks, when either collecting specimens or accepting specimens into the collection, do you look for anything in particular, unique features or special markings, etc.?
Yeah, so generally when we accept specimens for the herbarium, we want them to be reproductive. So we want the plants to have flowering parts or fruiting parts. We want them to be as complete as possible because again, if you think about specimens as being information, the specimen itself, the more you have, the better. For various kinds of uses you might have for the specimen. If you have flowers, if you have fruits, if you have leaves, the branch, and all that kind of thing, that makes them way, way more useful. And also, it allows them to be identified. A specimen that you can't really put a name on is not really super useful. And something else that we require is that all the information about the plant be present. When it was collected and where it was collected in particular. If we don't have that information, again, not really useful and maybe not really worth our while to keep it around because you can't really use it for that much. It's just too unknown. Yeah, so we do look for those kinds of things, the completeness of the specimen itself, the completeness of the data that comes with the specimen, that kind of thing. Okay, so let me move on and point out some other interesting things that we have while we still have some time. So I've been showing you these specimens that are in standard size sheets, packed away in the cabinets. But I alluded to something earlier, I think. These specimens date back hundreds of years. And the early specimens and the early herbaria that were around weren't really standardized like what we have here. They were a bit more free form, and they kind of were created, based on what the collector had around and what they wanted to do, what their whims were. And so what we have here is our 2 volumes of a collection of plants that were collected very early in New England. It's some of the earliest herbarium material that's out there. 4 specimens collected in New England. And this is a 4-volume set of specimens, nicely bound. So this person was a medical student here at Yale back in the early 1800s. As part of their medical studies, and I think also just because they were interested in natural history, they created a great collection of plants from the New Haven area. Over 600 specimens are in these bound volumes. And again, you can see these aren't the standard size. They're bound. They're not loose like the ones you've been seeing. But this is a very valuable collection nonetheless, historically and scientifically. You can see that this botanist, or actually this medical student, Horatio Finn, he took great care in putting this together. Very nice drawings. He made a title page. 1822 is when the plants were collected, roughly. And the plants, they still look great. He has little snippets of things here. He's got the flowers and fruits and the leaves and the branches. So they're complete specimens. He's got a scientific name. So we have a location, New Haven and environs. We have a year, 1822. We have a name for the plant, and we have the specimen itself. So these are really, really valuable. And I'll give you an example of that. There's a specimen here, which some of you might know. This is a purple loosestrife specimen. Now, this is a very aggressive, non-native plant that gets into wetlands and really takes over and alters the ecosystem and outcompetes all the native plants. It's not good to have this plant around. This plant was collected by Horatio Finn in 1822. And this is the earliest record that we know of in any herbarium. So I mentioned all these herbaria we have out there in the world with hundreds of millions of specimens. This is the earliest record we have of this invasive species in North America. So this is really valuable if you're studying how plant invasions occur, the history of plant invasions and how things spread. Because if you can understand that for the ones that are here, it might help you in the future to deal with things that might happen with new invasive plants, for example. So again, historically, quite interesting, this set of specimens, but also scientifically quite useful. So I'm just going to put that away. And while we still have a few minutes, I was going to show you, let's just move on down. I think we've got just a few minutes left. I'm going to move down and show you something else way down on this end of the herbarium. So I mentioned earlier that we have mostly flowering plants. Well, on this little aisle of our herbarium, this is where we have our fungi, we have our lichens, and we have our mosses, just on this one little aisle. But because mosses are stored in those packets and they're a bit more compact than the vascular plants, we actually have a lot of mosses here. Mosses in the broad sense. I'm including liverworts and hornworts when I say mosses. And we have we think about 65,000 specimens of mosses here, which is really quite good for a herbarium our size. It's really a large proportion of moss specimens. And among these mosses we have, they're from all over the world. They date back to the late 1700s, actually. And there's some very important specimens here. And I'll point one out or show one to you now. This is something I think that was maybe mentioned in the publicity for the talk. This is a specimen that was collected in Chile in 1834, I think. And it was collected by Charles Darwin. And so you can see the label here. This is not Darwin's handwriting. This is the handwriting of a curator that worked in the herbarium in the late 1800s, early 1900s, Alexander Evans. But you can see here, Conos Archipelago, Patagonia, C. Darwin. So here we have a liverwort. It's a kind of moss. collected by Charles Darwin when he was on the voyage of the Beagle. So really fun specimen if you're into natural history and biology and evolution. And there it is. So liverworts and other kinds of mosses, they're smaller plants than, say, most of your vascular plants, your typical vascular plant. And so this is actually a complete specimen, although there's not much material here. And so, a great, interesting specimen historically, also interesting scientifically. This plant was new to science when it was collected, and a new species was named based in part on this material here in our collection. This is a type specimen. I'm not going to go into what that means, but it's a kind of specimen that's required and very important in the process of naming a new species. And so Darwin collected this, it was new to science, and a botanist later named a new species based on the material. So historically and scientifically of great value. So maybe I'll pause for a second and ask if there are any questions. We're getting towards the end, I think, of the tour.
Okay, so Judith says, so good to see you in the herbarium again. How far along has the digitization process proceeded?
Hi, Judith. Yeah, Judith has a vested interest in this. Judith actually, if it's the right Judith, worked here in the herbarium as a volunteer and played a huge role in helping us digitize our North American mosses. How far have we gotten? We still have a lot to go, Judith. We've done all of our, won't go into the details. I'll tell you this, we have a new grant that started not long ago that's focused on digitizing our non-North American mosses. So you'll be happy to hear that we're going to finish our mosses within the next 3 years. So we'll at least get that part done. We've imaged most of our North American specimens, and we've imaged all of our type specimens. I mentioned type specimens earlier, and databased most of these groups as well. We still have a lot of non-North American material to do. So we still have tens of thousands getting into the 100,000 specimen range, I think, left. So quite a bit left to do. It's a slow process. Okay, I could take more questions, or I could show you another specimen if you want. It's up to you.
There's a couple questions. Sure. Chet asks, are specimens like flowers pressed for mounting, and how is that done?
I'm sorry, could you repeat the question?
Are specimens like flowers pressed for mounting, and how is that done?
Yeah, so right, so they are pressed. So nothing is really three-dimensional, with the exception being that sometimes people collect things into alcohol and keep them in jars. And so fruits and flowers and other plant parts on occasion are put in alcohol, and some herbaria have an alcohol collection associated with them. Here at Yale, we have a very small number of alcohol preserved specimens. We're primarily focused on the pressed and dried specimens. And it's just convenient to keep the plants that way. And believe it or not, you can, in some cases, and for many species, you can actually, if you needed to, rehydrate the flowers and study them if you wanted to have them not so two-dimensional. But yeah, they are pressed, flat, and dried. And actually, it's the whole plant often that is treated that way. So for example, if you were collecting a herbaceous plant and you could collect the whole plant, you'd pull it up, you'd get the roots, you'd have the leaves, you'd have the stem. And if it had flowers or fruit, you would have all of that together. And you would put that in some paper. Actually, we use newspaper. You put it within folded newspaper. You put that in a plant press, which is essentially like a sandwich of cardboard and absorbent material with a frame around it and some straps. You would make this big sandwich of specimens in newspaper, and you would put them in a dryer. It's low heat, as I mentioned earlier, and you dry them for a few days. And that's really the process. It's very simple. It's been what's been used for decades, centuries, I guess. Of course, if you don't have access to electricity, things get more complicated. If you're in the tropics, sometimes people will use like a fire. They'll use coals, and they'll actually dry their plants, under a tarp, say, in the field. Other times, they'll put all the plants into bags of alcohol and then bring them back and dry them later if they can't get to a plant press. But yeah, so the plants, the fruits, everything get pressed, flat, and dried.
Nancy asks, where do the botanical drawings fit in with the collection?
Where do they fit in? So we don't house the botanical drawings. We don't house drawings or artwork here. We have a few random things in our archives that were maybe drawings that an artist did when a new species was described or something like that by a faculty or staff member here at the Peabody. The drawings would fit well, one way they would fit in is that the specimens themselves would be the subject matter. And so, for example, if a new species is described, often there's a very detailed or a set of detailed pen and ink drawings that are done. And an artist would come in and consult with the botanist who's describing the new species, and they would look at specimens. They would pull specimens out of the herbarium, and the botanist would pick things that he thought represented, say, this new species that's being described. And the artist would actually look through a dissecting scope, for example, at high magnification. They would look at the plants that are in the herbarium, or these examples of this species, and make drawings from that. And so that's one way the art's connected to the plants. And they might even, as I mentioned earlier, rehydrate the flowers to make them more three-dimensional, more lifelike when they're doing their drawings, that kind of thing. And so there is that close relationship between art and botany, and many other connections as well. But in terms of housing artwork and that kind of thing, that would be the purview of an art museum and not really the herbarium.
Diane was wondering if you allow researchers to access collections.
Yeah, sure. That's a major function that we have. We do that in different ways. I mentioned earlier that we're digitizing our collection, so the things that go out online are very accessible and useful. But often, a botanist or whoever, an ecologist, evolutionary biologist, might need more than just what's online, more than a picture. They might actually need the specimen. And so when that's the case, they could visit, by appointment. We have people that visit and come in and use the collection. Sometimes it might be for a day. Sometimes they might come several days in a row. They might stay in town somewhere. Something else we do, which often people are surprised at, is we actually loan specimens between institutions. So I mentioned earlier, there are all these herbaria around the world. Many of them are well supported, like the one here at Yale. And we will do inter-herbarium loans, much like an interlibrary loan between the institutions. And when that happens, a researcher would visit some other institution. And at that institution, they would have access to our specimens. And we would loan the specimens to the other institution. They would house them there in cabinets, just like these. They would take care of them. The other herbarium that you're loaning them to would treat them as if they're their own specimens. And the researchers would come to that herbarium to do their work rather than having to travel maybe across the country or across the world to get to our collection. So that's another way we make them useful to researchers.
Laura was wondering if there were any special New Haven-based collections.
Yeah, right. So for example, I'll go back to the Horatio Finn collection. So these bound books I showed you earlier, that is the earliest, as I mentioned previously, it's some of the earliest New England plant collected material in the world. And it's all based around New Haven. So I'll just point out some more specimens from this collection. So here is another volume of this book. This is volume 1, Plants of New Haven. So these are all around 1822, collected right here in New Haven. And I mentioned, previously that invasive plant. Well, here's another plant that is kind of just the opposite. This is an orchid. Both of these are orchids in the genus Arethusa. This is Arethusa bulbosa, this specimen here. And this plant is a very rare plant in Connecticut, and it's actually not present in the area anymore. So this plant is locally extinct, you could say extirpated. And so we have a record of it having been in New Haven. And without this record, we maybe would not know that the plant ever occurred here. And of course, why did it disappear? We could speculate, land use change. This plant likes wetland environments. Wetlands are often drained for development, that kind of thing. And so presumably habitat destruction is what led to the disappearance of this orchid in the New Haven area and more broadly in Connecticut. So yeah, this is a very nice example of New Haven centered specimens. And something else I like to point out when I'm talking to people about herbaria, these plants are often collected for a particular purpose. Like in the case of the Horatio Finn collection, there was probably a medicinal botany focus with maybe a floristic component, someone wanting to know what occurred in the area. People who do classification and taxonomy might collect plants because they're describing new species and classifying life. If you're an evolutionary biologist, you might collect the plant to get DNA to understand its relationships with other species, that kind of thing. But once you have these specimens, they can have other uses, right? Horatio Finn maybe had no idea that purple loosestrife he collected back in 1822 was going to end up being this nasty invasive plant that wreaked havoc across the country, and that this plant would be important for studies of invasion biology. Another neat thing I like to point out, another nice collection of things I like to show people, we have a set of, we have some material that was collected by an anthropologist in the 20th century who was studying how people in peninsular Malaysia use resins from particular important trees that occur in the forest there. So you're talking lowland rainforest. And there's a very important group of trees in that part of the world called dipterocarps. And this anthropologist collected resins and leaf material and other parts of these dipterocarp trees for her study to better understand how they were being used by people who live on the peninsula. This is Malaysia again. But they have other uses. And so I'll show you these specimens and I'll talk about the other uses. And this is kind of an extreme case of an unexpected use. So here are these specimens, which are a bit unlike the other ones you've been seeing. They're in bags, and we have some leaf material here. We have some wood. We have these chunks of resin. So this is all resin collected from a particular kind of dipterocarp tree. And they have the name that people in Malaysia use for the plant, and something about the resin. It's soft. It doesn't get very hard. And again, the person who did this work, this was done for a doctoral dissertation back in the 1980s. This was important for their research. But while I was here at the Peabody a few years ago, I got a very interesting email from some people, conservators at the Getty Museum out in California. And they were like, they knew about Rosemary Gianos, that's the anthropologist. They knew about her work. And they were like, we hear that you have this great collection of resins from Malaysia. We'd be interested in acquiring some samples to do some destructive sampling, which we let people do if we have enough material. And you can see we have tons of resin here. So it's easy to give these people at the Getty a little piece of resin from certain species they're interested in. Excuse me. And they're interested in these species, in resin from these plants, because they were studying 17th and 18th century Japanese lacquer ware, so furniture. They were looking at the lacquers and the varnishes that were used on this Japanese furniture, and they were analyzing what the varnish or the lacquer was made of. And they needed chemical standards. So they needed knowns. They needed to know, they needed resin from things they knew the name of that they could get standards for to compare to things they would get from analyses of the varnish and the lacquer on the furniture, right? Because I'm not going to go into the details of the chemistry, but what it would do, it would allow them to identify the components of the varnish. They could figure out if this particular dipterocarp tree was used in the varnish, and that would then have conservation implications for them in preserving and conserving these furnitures at the different pieces of furniture at the Getty. So who would have thought, that these specimens would have that kind of use, in the future, and not really that far in the future. It was just like, 30, 40 years later that kind of use happened. So again, I hope I've demonstrated sort of why these collections are useful scientifically, from a conservation standpoint, and for other uses, art, history, so on and so forth. And again, we're just a microcosm of all these herbaria that are out there on the planet. And again, I like to think of them as sort of encyclopedias of plant knowledge. And actually the plants themselves, as you've seen, we actually have the plants. And so, really, really useful. And I hope that you come away with an appreciation for what we do and what a herbarium is and why they're important. And I think with that, we've probably reached the end. And I'm happy to take more questions if we have time.
Yeah, I think we have a couple minutes. So Jen asks, do you still collect specimens today, and how many per year?
Yeah, we do. So I didn't mention that. That's a very good question. And I usually bring that up when I talk to people about the herbarium. We're still collecting. People might have the impression that we have stuff that's old, but we do have things coming in up to the present. I've collected, with COVID, there wasn't as much collecting happening over this last year, but I collaborate with other researchers here at Yale. For example, faculty in ecology and evolutionary biology. And we study particular groups, and we still go out and collect specimens for evolutionary research. and also for and as a side effect of that, we're still finding new species. So in one group that we study, viburnum, which you all might know, viburnum is a group of ornamental plants. I studied that group with some researchers here at Yale. We go out and collect for that particular research, but in the process of doing that, we've collected new species. So we have at least 2 new species that we've discovered that are new to science that eventually will write up and describe as new species. So yeah, so people are still collecting for different reasons. People are still describing new species. There's still thousands of new species described every year on the planet, plant species. And other groups are even more, like insects. I think they're way more than that described each year. And even in herbaria, already collected, people have estimated that there are tens of thousands of sort of undiscovered species already sitting in a herbarium somewhere that people haven't yet discovered, if you will, as new species. So yeah, collecting is still happening locally and even internationally. And that's just here at Yale. You know, there are people, I didn't mention this earlier, but another component of herbaria are the people that work at them and the things that they do. And then collecting is a big part. We're still documenting what's on the planet, you know, where things occur, what they are, that kind of thing. And the people are a big part of that.
I think we have time for just one more question. Sarah was wondering if are there any descriptions on the specimen of what color the flowers were or what they did look like in nature?
Yeah, ideally that would happen. And it's kind of funny, you know, as you go farther back, there's less and less information on the labels. You might recall when we were looking at that old set of specimens from New Haven from 1822, there was just a scientific name and a year. And New Haven, it said New Haven and environs. It was like a general New Haven area. So that's pretty vague. As you go into the future, up to the present, people, collectors tended to include more and more and more information with their specimens. So now, yes, we include flower color. If there's a fruit that's kind of fleshy, we would include the fruit size even, and maybe the fruit, well, certainly the fruit color. If it's a tree, for example, you can't collect the whole tree, just maybe a branch. You would include the height of the tree, maybe how big the trunk was at breast height. That's a standard diameter at breast height. You would include things like that, things that you won't have on the sheet. And so, you know, you can't describe everything about the plant that's not captured on the specimen with the specimen. But, you know, people do get things like color, size, and maybe other interesting things they've noticed, like maybe abundance, you know, how much of it there is in an area, how rare it is, that kind of thing.
All right. I think that is all the time that we have today. Thank you to Patrick for giving us an awesome tour. Thank you to all you guys for hanging out with us and coming to this virtual tour. I hope you all had fun and don't forget to come to the tour next week, same time and day, entitled Getting By Without a Backbone, Peabody's Invertebrates. So thank
Thanks,
you all
everybody.
for stopping by. Yeah, thank you guys. Yeah, and I hope you guys have a good day.